Today we are going to study about the structure of protein. The structure of protein is divided into four types: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure. The primary structure of protein is relatively simple and consists of one or more linear chains of a number of amino acid units linked by peptide bond. Secondary structure. The conformation of polypeptide chain by twisting or folding is referred to as secondary structure. Two types of secondary structures, alpha helix and beta pleated sheet, are mainly identified. Indian scientist Ramachandran made a significant contribution in understanding the special arrangement of polypeptide chains. The primary structure is further divided into two types: alpha helix structure of protein and beta sheet structure of protein. Alpha helix is the most common spiral structure of protein. It has a rigid arrangement of polypeptide chain. The salient features of alpha helix are as follows. Alpha helix is a tightly packed coiled structure of amino acid side chains extending outwards from the central axis. Alpha helix is stabilized by extensive hydrogen bonding. It is formed between hydrogen atom attached to peptide nitrogen and oxygen atom attached to peptide carbon. The hydrogen bonds are individually weak, but collectively they are strong enough to stabilize the helix. All the peptide bonds, except first and last in a polypeptide chain, participate in hydrogen bonding. Each turn of alpha helix contains 3 to 6 amino acids and travel the distance of 0.54 nanometer. The spacing of each amino acid is 0.15 nanometer. Alpha helix is a stable conformation formed spontaneously with the lowest energy. The right-handed alpha helix is more stable than left-handed alpha helix. Here is a simple structure of a right-handed alpha helix. Beta pleated sheet. This is the second type of structure proposed by Pauling and Corey. Beta pleated sheets are composed of two or more segments of fully extended peptide chains. In the beta sheet, the hydrogen bonds are formed between the neighboring segments of polypeptide chains. The polypeptide chains in the beta sheets may be arranged either in parallel or anti-parallel direction. Beta sheets may be formed either by separate polypeptide chains. Hydrogen bonds are interchanged, or a single polypeptide chain folding back onto itself. Hydrogen bonds are intrachain. N-terminal, C-terminal. This is a parallel beta sheet. This is an anti-parallel beta sheet. Tertiary structure. The three-dimensional arrangement of protein structure is referred to tertiary structure. It is a compact structure with hydrophobic side chain held interior while the hydrophilic groups are on the surface of the protein molecule. This type of arrangement ensures stability of the molecules. Bonds of tertiary structures. Besides the hydrogen bonds, disulfide bonds, ionic bonds, hydrophobic interaction, and van der Waals force also contribute to the tertiary structure of proteins. Quaternary structure. A great majority of proteins are composed of single polypeptide chains. Some of the proteins, however, consist of two or more polypeptides which may be identical or unrelated. Such proteins are termed as oligomers and possess quaternary structure. The individual polypeptide chains are known as monomers, protomers, or subunits. A dimer consists of two polypeptides, while a tetramer has 4 polypeptides. Bonds in quaternary structure. The monomeric subunits are held together by non-covalent bonds, namely hydrogen bond, hydrophobic interactions, and ionic bonds.